everybody, and welcome to the Informatics Institute PowerTalks seminar series. I am very happy to introduce today's speaker. It's very well known to us, Dr. Wayne Liang. He's Assistant Professor of Pediatrics at Emory University. He's a physician informaticist and pediatric hematologist, oncologist, and bone marrow transplant physician at Cancer and Blood Disorders Center at Children's Healthcare of Atlanta. Very previously, he was assistant professor at University at UAB in the Informatics Institute right here. So I feel like you just never left. And it was also a pediatric hematologist, oncologist, and bone marrow transplant physician at Children's of Alabama. He completed his bachelor's in anthropology and also his MD degree at Case Western Reserve University, his residency in pediatrics, where he also served as chief resident at Vanderbilt University, a fellowship in pediatric hemonc at Seattle Children's, and also an NLM postdoctoral fellowship and Master of Science at the University of Washington. And the name of his talk is Development of Computable Pediatric Chemotherapy Regimen Knowledge for Cancer Clinical Care and Research. Dr. Liang, my great pleasure to introduce you.
Thank you so much, Amy. I'm so glad to be back visiting with you all, my former colleagues at the UAB. I'm happy to give the talk today. As Amy described, I'll be discussing the development of computable pediatric chemotherapy regimen knowledge for cancer clinical care and research. And this is work that was initiated while I was still at UAB and is ongoing work. And so first I want to disclose provide my disclosures. I have a paid consulting relationship with WISDAT to develop pediatric chemotherapy regimen knowledge for Hemonc.org in collaboration with the National Cancer Institute Surveillance Research Program. So the contents of my talk today will be addressing the nature of this work. I don't stand to benefit from any sort of licensing agreements or whatnot, given that the work, the knowledge that we're creating is openly available.
Hello.
And if you are not muted, can you make sure that you mute yourself? Thank you. All right, so here is our agenda today. I will provide some background on pediatric cancer treatments and the problems that we encounter in this space, particularly as it relates to understanding the data behind it and how regimens are developed. I will then discuss the National Childhood Cancer Registry, for which this work is being conducted. And then finally, at the end, I will describe our progress with developing computable cancer regimen of vocabulary. So pediatric cancers are rare diseases. And the reason why I say rare diseases is because cancers are different from each other. So as a group, worldwide, of all cancers, that are diagnosed, 90 million people are diagnosed with cancer, of which only 1 to 2% are in children. In the United States, about 16,000 children are diagnosed with cancer every year, and that comes out to an incidence of about 1 out of 285, which is about 0.3 to 2.4%. So that's all pediatric cancers of all subtypes. So pediatric cancers as a group are rare, and each pediatric cancer type, specific type, is very rare. However, despite their rarity, pediatric cancers are life-threatening, and they make up a large proportion of pediatric deaths. In fact, they're only second to accidents or unintentional injuries as a cause of death in the pediatric population. So particularly for children under 15, you can see highlighted here. So rare disease, but deadly disease. When we talk about cancer treatments, there are basically 3 classes I like to think about. I'm a lumper, so I like to make things simple. You either cut it out, or you fry it, or provide energy to it, or you give medicines that treat the cancer. And for all these regimens, they're all harmful to the body, but the idea is to harm the cancer or kill the cancer without harming or killing the patient. And in our world, the cancers come from the patient. And so almost all of our treatments that are harmful to the cancer are also harmful to the patient. And so there's an art there to try to develop a regimen that's sufficiently toxic to the cancer without being excessively toxic to the patient. So when we talk about cancer treatment regimens, what we mean by that is some basic vocabulary. When we say chemotherapy, we mean any drug, any medication that is active or toxic against cancer. And as I described, it's also toxic to the person, to the patient. They work through a variety of mechanisms. And so when we create cancer regimens, we often combine different types of chemo that uses different mechanisms in a combinatorial way so that we can minimize the toxicity of each individual agent. Because you can imagine, as you increase the dose of a chemotherapy agent, the toxicity also increases. But what if we can give a medium dose of this agent plus a medium dose of this agent plus a medium dose of this agent? Hopefully with a combination, it would be sufficiently toxic to chemo without hitting that really high toxicity of each individual agent. And that is one of the theories behind why we use combination chemotherapy to treat pediatric cancers. And so when we talk about treatment regimens, we're basically talking about a recipe containing ingredients put together in a certain way, just like you may have a recipe to bake a cake or cook a dish. This really toxic recipe of chemotherapy agents and surgeries and radiations and whatnot, given at specific intervals, at specific doses, at specific times, to provide an outcome, a desired outcome. So here's an example of a real-world cancer regimen. Children's Oncology Group AHOD 0031, used to treat Hodgkin's lymphoma. And you can see here's an example of what a recipe might look like for a cancer treatment. The recipe, we call it a shorthand ABVEPC, which is short for these drugs. I don't want to bring attention to the fact that we use here Adriamycin, which is a brand name, and for doxorubicin to make the ABVEPC. And you can see here that this chemotherapy agent or regimen contains these chemotherapies at the doses outlined below, given over a certain amount of time at certain days. So you can imagine a calendar of sorts, right? On day 1, you receive these agents. On day 2, you receive these agents. On day 8, you receive these agents. And then prednisone, which is an oral drug, is received days 1 to 7. So 1, 2, 3, 4, 5, 6, 7. And then the next line under that is the cycles. So we'll give this at 21-day cycles for 2 cycles. And then we evaluate the response and give them 2 more cycles. So 2 21-day cycles, and then no further treatment. So a cancer regimen might be one cassette that's repeated multiple times at some interval. And this is a fairly simple, a straightforward regimen that you might encounter. And this is pulled directly from Hemon.org, which I'll be talking about later. Another example of another cancer treatment regimen. This one's fairly complex. So complex I can't really show you all of it by screenshots, given that it just doesn't fit on the screen. The COG, Children's Oncology Group AALL0932 protocol, which is a protocol for treating childhood B leukemia, standard B leukemia, contains several different phases, including induction, consolidation, intermaintenance, intensification, intermaintenance 2, and then some type of maintenance arm. And this is given over a course of about 2.5 years. This is a treatment protocol. There are different arms that are outlined here. So each of those named phases are different from each other. So for example, this is induction. And you can see that it has these drugs outlined here, PEG, asparaginase, vincristine, dexamethasone, and some type of CNS prophylaxis. I didn't outline all of it here. It's given on some interval, days 1. PEG asparaginase is given on day 4. Vincristine is given on day 1, 8, 12, 15, and 22. Dexamethasone is given every day from day 1 to 28. And then CNS prophylaxis, which are lumbar punctures with chemotherapy, are given on certain days depending on patient characteristics. So that's only induction. If you're talking about consolidation, it's something totally different, a totally different recipe. This recipe must follow induction. And then intensification, again, must occur at a certain time point with different recipe altogether given certain time points. So that's what a regimen might look like. And again, this is a real regimen pulled from hemonc.org. We can already kind of see the complexity that might be involved in understanding the regimen that a patient received. So how are cancer regimens developed? Well, there's a balance here. We're balancing the fact that it's a disease, so we're trying to cure patients. Also, we want to try to prevent them from having long-term side effects that causes long-term morbidity. So 1 out of 8 children with cancer will not survive. So if you start with cancers with poor outcomes, our strategy is to then escalate intensity. And when I say escalate intensity, I'm saying increasing dosage or adding more chemotherapy agents, adding more cycles, just adding more and more things on, more toxicity to try to get the desired outcome that we would like. And we know we hit the limit when it's excessively toxic. So if you have a clinical trial where too many patients die, you say, okay, I got to back off and we can't use that regimen. Or you get the response that you want. You get to a certain level of cures and a really certain number of, unfortunately, acceptable number of lack of responses that you think is sufficient balance with the toxicity. So that's the escalation phase of regimen development. On the other side, when we want to prevent long-term side effects, we also decrease intensity. So that's what's happening right now in standard risk leukemia, where we have achieved for a subpopulation of patients really good outcomes with survival rates of cure rates of greater than 96%. Yes, 4% are not cured on the front end, but for 96%, we're now able to decrease the intensity of treatment for that subpopulation. And then we know we hit the right intensity when you go one step below and your outcomes are not what you desire. Things say, okay, that's not going to work. We're going to go back up to the prior intensity. And it's thanks to the patients who are willing to enroll in clinical trials who are putting themselves out there for us to be able to discover the ideal regimen for each type of cancer and cancer subtype. So the development of cancer regimens involves traditionally clinical trials. And this is, as many of you are aware, a very time-consuming, a very expensive process, starting from preclinical discovery and then regulatory approval to then all the different phases of clinical trials, which I won't go into in detail, and then FDA approval, and then publication, or actually in between, publication of the evidence. So you have to wait for the evidence to mature before you can publish it, and then FDA approval. So the speed of cancer regimen development and therefore improvements or cure rates in patients is limited by speed of running clinical trials, the evidence life cycle, if you will. It has worked. So over decades, so to understand this graph, these are decades, and this on the y-axis is survival rates. So it's good. And you can see for each cancer type that we're dealing with, it's an overall trend, as you can see with this green arrow, of rates going up. And so for Hodgkin's lymphoma, going from 40% survival in the 1940s to now almost 100%, 95% all takers. So all cancer types in general have improved in their survival, but not in an even way. You can see here AML started off with 0, and now we're achieving about 50 or so percent. This is in 2000, so this is 20 years ago. The trend holds that the survival rate is still around here. And so this kind of illustrates the fact that pediatric cancers are a disparate family or group of diseases that are different from each other, and we're not yet achieving success in all fronts. For these type of cancers, we can start decreasing toxicity for certain subgroups, and for these, we're still escalating and hitting sometimes maximum toxicity. Other thing I want to outline here is it's taken decades to improve it. And there's really a desire to accelerate evidence generation cycle so that we can improve cures faster. So the discovery life cycle is slow, right? As I mentioned earlier. Also, because pediatric cancers are rare diseases as a group, and then even rare diseases individually, we have small n's. And with small n's, it's difficult to enroll the number of patients required to generate the evidence base you need to be able to find that one regimen is better than another. For leukemia trial, you can probably enroll enough patients across all the major centers around the country in a couple of years to maybe ask one question. But if you want to ask 4 questions on a clinical trial, you got to enroll even more patients over time. And, even though, unfortunately, cancer rates are increasing among pediatrics, that's not really a strategy for how we're going to enroll enough patients on a clinical trial. It's not enrolling that fast. So basically, we're limited by the amount of time it requires to enroll the number of patients required to adequately power a study. And if you're talking about a super rare condition, it may not be possible to run a clinical trial. There may be no clinical trials available because you might only enroll, you may only have 10 cases over several years. And then representativeness. This is a major issue in adult oncology, more so than pediatrics, where enrollment is much lower. In pediatrics, our overall enrollment in clinical trials is about 50%. But again, it's very uneven, much higher in common cancers, much lower in cancers in which there are no clinical trials available. And we also all know that conditions within a clinical trial are different than conditions in the real world, because those who are eligible or decide to sign up for a clinical trial are different than the overall population of patients who receive that regimen in the real world. So the question is, are there alternative sources of evidence we can use to augment the evidence that's generated in clinical trials? Can we use other sources of evidence to accelerate the discovery life cycle, even if it's not ideal types of evidence, than the standard randomized control trial? That's where real-world evidence comes in. Real-world data is data that's generated from a variety of sources, usually outside of the context of a traditional clinical trial. And real-world evidence is evidence analysis or evidence that's generated from analyzing real-world data. And so what do I mean by that? Well, here are some examples of real-world data that could potentially provide evidence to augment clinical trials data. Clinical data are EHRs, potentially. You can look at what real patients in the real world, what treatments they're already receiving and what their outcomes. Of course, the data that's generated in real-world data is of a different quality than randomized clinical control trials. But in a setting where you have limited data available, it might be worthwhile to use that at least for hypothesis generation. That can be later validated in another setting. But we should think broader than just EHRs. Medication data may be containing EHR in the medication administration record. If you write prescription, data lives actually somewhere else. It lives in the pharmacy data, in CVS, Walgreens, things like that. And you can also look in claims data to look at, did they actually fill that chemotherapy agent that I wrote? How often they're filling it. Does it indicate that they're actually not taking the medicine regularly? Things like that. So that's an example of real-world data. And here, claims data, molecular profiling data, family history data, even social media data, environmental exposure data, where do they live in relation to the interstate? Is the toxins that are being released in the air from an interstate related to cancer rates, or local radon or other sort of environmental factors? You can start imagining that if we're able to tap into real-world data, it really expands the evidence base through which we can potentially make discoveries to advance cancer cures. Specifically, I'm interested in, can we identify treatments that patients in the real world are receiving and compare them against each other, particularly patients that are not in clinical trials? So one source of cancer data that I want to focus on is cancer surveillance data. So cancer surveillance is a legally mandated public health activity conducted to collect and analyze cancer-related data. There are 2 major cancer surveillance programs, public cancer surveillance programs in the United States, that are complementary to each other. One is the NCI, the National Cancer Institute's SEER program, where they identify target geographic areas in the country where they collect data at those locations to try to cover about 50% of the US population and then make some assumptions based on that 50%. Another complementary approach is the CDC's National Program for Cancer Registries, which supports state-based cancer registries, for example, at the Alabama Department of Public Health, or each state in the Public Health Authority may have a registry. Not all states have it, but most states do, and this program covers about 96% of the US population. They do collect slightly different data, and so often the data is merged to provide cancer statistics like the ones I provided earlier in the presentation. So how does cancer case reporting work? Well, first, a patient with cancer presents to a healthcare organization, and they're diagnosed there and they receive treatment there. Without going into very much detail about who gets counted by healthcare organization, I'll move on now to the next step. The healthcare organization is now legally mandated to report cancers that they're required to report. What I mean by that is not all cancers are reportable. Only certain ones are reportable. So the healthcare organization has identified the subset of cancers that are reportable. And then once they identify those cases, then they abstract the details. So usually that's performed by a certified tumor registrar, a staff member, from the EHR into specialized software like Metric, for example. And this is often a very manual process, possibly semi-automated, but largely manual, chart abstraction, chart review. These cancer registrars are trained in identifying cancer concepts, but they're not clinicians. They're not oncologists. They're not nurses. So they're going by what is written in the record, and then classifying it based on terminologies, just similar to what a medical biller might do in reading a record and then billing. And then it's submitted oftentimes to a centralized registry through an online web portal. Oftentimes, maybe there's an upload of a file or manual entry. And then what does that centralized cancer registry do? Well, what it does is receive the data, aggregates it, cleans it, analyzes it, and then usually several years later releases the data after all this has occurred. So that's cancer surveillance. So I do want to mention now the work that Dr. Osborne, my former collaborator at UAB, is doing in this space using natural language, machine-mediated, often natural language processing mediated identification of cancer cases, and particularly reportable cancer cases, to help the cancer registrars identify who they need to abstract. So cancer registries as a form of real-world evidence has some challenges. One, it only includes reportable cancers. Cancers that are not reportable are not counted. It often relies on human abstraction. I'm not aware of any fully automated methods. At very minimum, they're all human involved. It's a fairly manual process. The data quality, is variable. As I mentioned, these registrars are trained, but they're not clinicians. And there is uneven availability of data standards. For example, data standards are fairly available for medications, or there's a pretty well-established data standards for tumors, identifying tumors and the grading and staging. But there lacks a lot of data standards in other areas that are relevant to cancer care. Of note, the treatment data in cancer registry data is very limited. Oftentimes, only the first course of cancer treatment is included. As I described earlier, cancer treatment can be very detailed and if the first treatment fails, you might give multiple courses of treatment afterwards. But the registrar is only required to abstract the initial treatment. And so oftentimes the treatment data there is quite lacking. And it's not linked to other data sources. You only have the data that you collect. If you don't collect it up front, you don't have access to it. And so if you don't collect all the treatment data, it's difficult to perform analysis on outcomes, for example, for patients based on the treatments they received, when only you have the first treatment that they received. So the National Childhood Cancer Registry is a new effort launched in the last couple of years to improve upon and modernize the U.S. cancer registry infrastructure. The idea is to provide a centralized data resource linking all existing data for all cancer patients below the age of 30, up to 39. So that includes pediatric and adolescent and young adult identified as less than 40. And it's a very ambitious goal. The thought is that using the cancer registry data as a foundation to now link out all different data sources on the same patient so that you can now describe the complete trajectory of a patient's treatment from diagnosis to initial treatment to survivorship versus relapse to long-term side effects to the health outcomes 40 years from now, for example. And so the vision is to take existing cancer registry data, link them to detailed clinical data, and ideally data exported directly from the EHR. Think about other data types like radiology data, reports and images, genomic data, survival data from other vital registries or vital records. As I mentioned earlier, pharmacies, insurance claims, and other real-world data. Now, this I want to really stress is this is the vision. Infrastructure is not built yet. Right now, the NCCR has requested each clinical site that are participating in the pilot to provide data in whatever format they're able to provide. They're saying, we want to expand the data collection effort of cancer registries. We want to see what data is available in your center. Send them all to us, and then we're going to sort it out. And so right now, the process the NCI is going to in this program is now looking at all the different data submitted from multiple different institutions of without semantic interoperability to understand how they're going to make sense of it and of how they're going to be able to ingest so many different data types into this modern infrastructure. So this is ongoing. There will be an RFA put out soon for someone to be a data coordinating center or data provider to build the infrastructure. So that is something that is not awarded yet, to my knowledge. So that's where we are to get to this vision. Now, this is a wonderful vision, and I think we all would love it if this can happen. But there's many, many challenges. I just want to show you a little bit of the quote-unquote architecture of the database here, using registry data as the foundation, and then now all these other data sources we want to link in to try to integrate a view of the patient, and then make this database, or not really a database, but this federated infrastructure available to researchers who want to understand cancer. I mean, in many ways, it's similar to data commons work that's being done at UAB and elsewhere, being able to integrate different data sets, essentially. But in a federated model, not centralizing all data in one place, but be able to link out to other resources. So the ability of this data infrastructure to answer questions is limited by the quality of data that's already in it, right? Garbage in, garbage out. So existing cancer registry data, as I mentioned, is limited and also not standardized, especially when it comes to treatment data. A registrar may write down the drugs that the patient received, or they might write down the name that the oncologist wrote down as a regimen name. Like my prior example, I might write ABVE-PC, and that might be what the cancer registrar abstracted. But there is no standardization for regimen names and the components. And that ABVE-PC example is good because there is actually more than one ABVE-PC regimen variant. They're actually quite different, not quite, but they're different in dosage, and they're given to different subclasses of patients. So the name of the regimen is the same, but the regimen is actually different. And because there's no standard, this treatment information is collected in a variety of different ways within existing cancer registries. So what if we can integrate? Like if we can identify a patient in a cancer registry, and we can pull in their clinical, their chemotherapy, their medication data from an EHR, Can we integrate that? Well, that's yes, that would make the data richer, right? Definitely. But there is, besides challenges with semantic interoperability, for all these different EHR types and whatnot, there's that foundational problem of lack of a vocabulary. Even if we get all of this treatment data, we have chemotherapies, when they were given, whatever, how do we know what regimen they received? The same applies to other real-world data sources. So that is the reason why we're working on developing a computable cancer regimen standardized vocabulary. The vision, to simplify it, is that if you're given a cancer regimen, you can provide cancer regimen details. And vice versa, if you're providing cancer regimen details, you can identify with some level of certainty, there's going to be some probability there, of the cancer regimen that was received. You can imagine if a case says they received A, B, B, E, P, C, I can at least identify the drugs that they got. And then if I have a patient who received these drugs, I might be able to match them using various different methods with a regimen that contains those ingredients. How that relates to research being done using centralized cancer registries, or just the work that is being done, is, if we support a vocabulary upfront, then a registrar can just select the right regimen upfront in the data. Garbage in, garbage out. So improve the data quality going in, you'll improve the data that's coming out. And for existing data or data where that is missing, if you have the treatment data, the specific details, you might be able to identify the regimen that they received. And if we're able to flesh that out, combine that with outcomes data, vital statistics data, you can now imagine a world where we can perform comparative analysis. Let's compare patients who have this type of cancer, who receive regimen A in the real world versus regimen B in the real world versus regimen C in the real world, regardless of whether or not they receive it on a clinical trial. That is the vision we're trying to work towards. Let's look at all patients with cancer in the United States and all of their treatments. Give me all the data. And as everyone knows, that is a vision that is very difficult to achieve, but that is what we're working towards. So Curtis asked a question about regimens. There's no national repository of all the regimens in the world, or all the regimens being used. Regimens, if they come from clinical trial, yes, they're documented. Oftentimes they're registered in clinicaltrials.gov. Clinicaltrials.gov is a listing of protocols, or rather clinical trials, which is related to the protocol that they receive. Clinicaltrials.gov itself does not contain all of the regimen information that is given. And then secondly, the regimen that's given in the real world may differ from the regimen that was studied on the clinical trial. So I don't know if that answers your question, Curtis. If there was more time, I can go into that some more. So this is what we've been working on. So Heman.org is a website. You can go to it right now if you like, literally Heman.org. It's basically a Wikipedia of cancer regimens. This was founded by Peter Yang and Jeremy Warner, who are two adult oncologists. They started during their fellowship, oncology fellowship, as a shared physician notebook. I'm learning about cancer regimens. I'm going to be writing these down anyway. Might as well share them with the world and make it a public resource. And it since then has grown from 2 people to now many oncologists involved in this effort. It is now the largest freely available medical wiki of cancer regimens available. As I mentioned, founded in 2011 and curated by an advisory board, or an editorial board of board-certified hematology, oncology physicians, and pharmacists. And as of today, it contains 3,850 regimens and more than 5,000 variants of those regimens, linked to 5,900 clinical trials, and with this many references, we're linking to the publication references that regimen. And it's primarily adult regimens, given that it started among adult oncologists, but growing pediatric regimens. So I've been involved with this effort for several years now, 1, 2, 3, 4, 5, 6, at least 6 years. initially as the only pediatrician in a group, and thankfully now we have grown that to be able to increase it. So the hemonc.org website then is the foundational source for the hemonc vocabulary. The human vocabulary is a standard vocabulary of chemotherapy regimens representation in an OMOP common data model. And so this vocabulary is derived directly from the website through the use of a parser that reads a wiki syntax. And this vocabulary is free for non-commercial use. So you today could use this in your research or in your application, as long as it conforms to the licensing agreement that is set forth. There are two places that you can access this vocabulary. One is within Odyssey Athena, which is a vocabularies repository. And so when it was contributed to that, it was the first ever hemonc oncology regimen vocabulary contributed. And that's where I believe the only. And also, if you want to download it, you can register for a free account at the Harvard Dataverse, a repository of data, and download it yourself, export the data. Again, that is available for free for non-commercial use. And the treatment regimens on the website so the website is written in a human readable format, but with a syntax using wikis headers and whatnot for specific fields. And then when it is parsed, is now in a structure format that is machine readable, if that makes sense. The problem that, John, you might be illustrating is that for real patient data, that data may not be captured. I mean, ideally, we would capture this data upfront in the EHR in a structured format linked to a vocabulary. That is not in existence right now, but that's certainly possible with this resource. And Curtis, yes, this is primarily on physicians' free time. So here's the example of the Dina browser for the browser vocabulary, and here is the Dataverse where you can download it to your own workstation to explore. So this is the pediatric team involved in abstracting pediatric regimens. So we are working in partnership. The NCI identified this as the vocabulary they wanted to support, given that it was the largest and openly available vocabulary of cancer regimens. So they, in partnership with WSTAT, asked us to focus on abstracting pediatric regimens. So I was able to build a team from only myself to now 3 physicians, myself focusing on pediatric leukemia and lymphoma, Dr. Nikki Wood at Mercy Children's in Kansas City, who is now section editor of pediatric neuro-oncology and solid tumor, and Dr. Chavni Gargan, who is the section editor for pediatric sarcoma, also currently at Mercy Children's in Kansas City, and soon to be heading to South Carolina. And we're all board certified or board eligible, and providing medical oversight for regimen abstraction. Supported also by Austin Fitz, who is a pharmacist and a pharmacy resident doing focus abstraction, and also Karan Patel, who is a medical student, who's also learning how to read regimens and abstract them. This is our workflow. First, we identify candidate regimens to abstract. We then prioritize them. So we prioritize what is currently accepted standard of care. If it's currently being used in our field and that is standard of care, we're going to abstract them as long as there's evidence behind it. Sometimes standard of care regimens don't have evidence basis, unfortunately. We prioritize those broadly used in the last 10 years for recency purposes, and that's also per request of the NCCR, given that they are focusing on registry data from the last 10 years. And we use some data to help us identify which ones to focus on. For example, we use our own local institutional order set usage to identify regimens to abstract. And we do want to include regimens on clinical trials that were the negative arms, given that these patients who are reported will have received those regimens, even if they're now discovered to be inferior to other treatments. In the future, we would like to actually use existing registry data to help us identify regimens that we should abstract. We prioritize regimens that has published evidence, given that we want our website to be useful for practicing clinicians as a quick reference of what regimens they could give. We then identify the supporting evidence. Usually is a publication of a sort. Sometimes it's an abstract, unfortunately, but sometimes abstracts are practice changing. So if it's practice changing, we include it. Sometimes supporting evidence is in like a lower tier evidence source, but and then we identify sources of regimen details. Unfortunately, when results of a clinical trial is published, they don't always include the protocol. And sometimes they don't include even just the details of sufficient details of the regimen to recreate it. It's as if you publish a study, but your method section is not recreatable. Unfortunately, that's a reality right now in clinical trials world, where we publish, this regimen is fantastic, but you don't actually provide like the recipe for how to give it, how an oncologist can actually give it. And that makes it challenging for us, given that we want to abstract regimen details. And we want to respect that protocols are often proprietary until they're published. And so this is always a fine line that we're walking, identifying what's the most appropriate source from which we will abstract regimen details. We then abstract it, and then the parser at some batch intervals will then parse it and then pull into the vocabulary, which is then contributed at some interval multiple times a year into the Dataverse. So our progress to date, since April of last year, we were able to migrate 12 existing pages to the pediatric section of the website. We have abstracted 25 regimens across pediatric leukemia, lymphoma, sarcomas, brain tumors, solid tumors. And these regimens include chemotherapy as well as radiation, or if a radiation is included in the regimen. We continue to have ongoing development of the structure of how we abstract these details. It's still not perfect, so it's a work in progress, just like a wiki is. But we're continue to iterate to improve it. And then we're continue to work closely with the NCCR at the NCI to discuss how can we integrate our resource with the future NCCR platform that they will build. So that, for example, if a registrar is using their platform to abstract, identify a case and validate it, they can then classify a patient under a certain regimen in a vocabulary. That is the vision. And some of it is limited by, first of all, the platform doesn't exist right now for NCCR. And two, we're using MediaWiki software, the same software that underlies Wikipedia, and so we're limited by the functional limit, the programmatic limitations of that platform. Some challenges that we continue to face, is that pediatric cancers are rare diseases, and therefore some of the evidence that we would like is just lacking compared to adult trials, where you can easily enroll the number of patients you want with just a few sites. We're not always able to do randomized controlled trials. Sometimes we have to do a single arm trial to enroll enough patients and then compare with a historical cohort and just assume that they're similar enough that we can do a comparison. And we often do basket trials where there are multiple comparisons in a single trial because, again, the number of patients are limited. Often, we will get preliminary data that's shared with a clinical trials network, like a children's oncology group, that's not yet published, but because it's shared informally, it's immediately practice changing. So we struggle with the practice has changed. This is not how we treat this cancer anymore. But they haven't published it. And so do I abstract it? That is a challenge. We're dealing with proprietary nature, even though some of these clinical trials networks, like the COG, is publicly supported by the NCI, the protocols themselves are still proprietary. And there's also some safety concerns about making protocols available when the evidence is not yet there that treatment is beneficial. So you can imagine if you don't understand that and you give a patient a treatment that has no evidence and they have a bad outcome, well, maybe you're treating them prematurely with that regimen. I just want to quickly highlight some complementary efforts. So the Pediatric Cancer Data Commons, which is led by Sam Volchenboom, one of my one of my mentors as well, is doing similar data harmonization efforts, but in the clinical trials data space. This effort is trying to be the central repository for all pediatric cancer clinical trials data in the entire world. And they want to harmonize all the data so that you can ask questions across clinical trials, across clinical networks, across continents even. How does a pediatric patient with pediatric leukemia do on a CLG protocol versus a St. Jude protocol versus a German protocol versus a Japanese protocol? And that requires getting all the data in one place, and that requires harmonizing the data dictionaries, which is no easy task. But some of the data elements that they are harmonizing are the same data elements that we will need to harmonize on the cancer registry front. And then also there's a Center for Cancer Data Harmonization, also led by Sam Wilchenboom, that is trying to standardize what the vocabularies that are used in all these different cancer-related data. So in summary, today I reviewed the problem space with cancer regimen development and the problem using real-world data to generate evidence to improve our cancer outcomes. And I reviewed the development of the National Childhood Cancer Registry and our work with Hema.org to develop a computable cancer regimen vocabulary. And thank you so much for your attention. And now I would like to open up for any questions that folks may have.
Thanks. Great presentation. Actually, there are a few questions in the chat if you want to address those, or if you don't see them, I can read them too.
So I've addressed most of the initial ones. Unless there are additional questions or additional parts of it I didn't address, please go ahead and put them in. Let me address one from Hope Gray. Are adverse reactions tracked for various cancer regimens? So yes. So on a clinical trial, we're required to track adverse outcomes and adverse effects or events, and then make a designation whether or not we think is related to the regimen that we gave, and specifically is related to the study drug. And all of that is reported to clinical trials. So all the data is contained within clinical trials data. Unfortunately, the reporting requires manual identification. A human PI identifies that an adverse effect has occurred. And then a human clinical research associate then submits that to the network. And work done by Tamara Miller here at Emory has shown that the report rate is around 50%. And so a lot of the adverse outcomes or events are just not being captured in clinical trials. So what she's working on with investigators at Children's Hospital of Philadelphia is to create they have created a adapter that you can implement in your EHR that identifies likely adverse events that has occurred, that can then pull that into a REDCap registry that can then be used to submit with your clinical trial. So machine-mediated identification of a phenotype, of an adverse outcome is being worked on. Looking at the real-world evidence side, this is what the FDA wants to do now, is after a drug has been approved, to do ongoing tracking, monitoring, and of course you can rely on people reporting adverse outcomes, or you can do other ways of identifying, using EHR data or looking at Twitter data or whatnot to try to identify adverse events associated with a drug. So that type of data is envisioned to be part of the NCCR once it is created. Thank you. Jim Semino, do you run into problems with drug data that are blinded? So with what we're working on, we're particularly working on the regimens. So we're not working with the outcomes. So that is an issue, but it's not directly related to what we're working on. However, relevant to that is that drug data that's blinded. So, like for example, a randomized trial where there's blinding of who received the drug on RMA, like a double-blind situation. Well, it still does not affect our work directly because we can abstract the regimen. Now, it's a very good question in terms of how that data is reported. If you're using real-world data, the EHR may not contain the arm that the patient receives because it's blinded, right? So that is a really interesting problem. If you can, if you have the clinical trials data available, and you have the EHR data, and you can identify and link individuals, then you can imagine that the clinical trials data will have the code of who received the treatment, who didn't, even if the EHR doesn't contain the data. So I imagine that's how you might want to address that problem. The major problem here is identifying who is who, given that this is at the end of the day for a researcher, when they receive the data, is a de-identified data set. The clinical trials data is also a de-identified data set, although there are mechanisms that they're trying to develop. Sam's group is trying to develop to try to be able to identify a unique individual within multiple data sets. But we're all aware that there are many problems with identifying unique individuals and then linking them. Thank you for the question.
Any final questions? We have about 2 or 3 minutes left. Well, if not, then you can always contact Dr. Liang later to send in your questions. Thank you for the great talk, very informative, and the recording's available if anybody wants to access that later. And thank you for everybody for attending, and have a great weekend.
And I want to second that. Wayne, thanks very much for coming to talk to us. We miss you, and glad to see that you're thriving and combining real patient care, real medical research, and informatics. So that's terrific. So thanks very much for contributing to our seminar series.
I'm so happy to be able to visit you guys virtually. Unfortunately, not in person, but at least virtually to see you guys, and miss you all very much as well at UAB.
Great.
You're not that far away, so hopefully next time we'll be in person.
That's right.
Bye, everyone.
All right, take care. Have a nice weekend.